All right, guys, welcome to the Chapter 17 lecture about blood gases, pH, and buffer systems. Now, we've broken down Chapter 17 into two parts because it's a pretty complicated chapter. So what we're going to cover in the first part is kind of what the blood gases consist of, how pH is buffered, what that physiology looks like, and then we will move into part 2. I'll be posting that at the beginning of this week as well, probably tomorrow or the next day. And that will cover more whether someone is going through respiratory acidosis or metabolic acidosis or alkalosis. So it'll be more what you're going to see as far as the lab work goes, and then being able to surmise a diagnosis from that. Okay, so let's just go over the objectives that we're going to cover today and in the next lecture. So after completing this chapter, you should be able to describe the principles involved in the measurement of pH, PCO2, PO2, and the various hemoglobin species. You should be able to outline the interrelationship of the buffering mechanisms of bicarbonate, carbonic acid, and hemoglobin. Explain the clinical significance of the following pH and blood gas parameters: pH, PCO2, PO2, actual bicarbonate, carbonic acid, base excess, oxygen saturation, fractional oxyhemoglobin, hemoglobin oxygen binding capacity, oxygen content, and total CO2. You should be able to look at data and determine whether it's normal or represents metabolic or respiratory acidosis or metabolic or respiratory alkalosis using the Henderson-Hasselbalch equation and blood gas data. Then from there, you should be able to identify whether the data represents uncompensated or compensated conditions. This is what we're going to cover in the next lecture. We won't be covering this quite yet today. We're going to identify some common causes of non-respiratory acidosis and alkalosis, respiratory acidosis and alkalosis, and mixed abnormalities. Then you should be able to state how the body is going to attempt to compensate for that using the kidney and lungs for various conditions. Describe the significance of the hemoglobin oxygen dissociation curve and the impact of pH, 2,3-DPG, temperature, and PCO2 on its shape and release of oxygen into tissues. And given a set of arterial blood gases and pH results, determine which acid-base disorder is most appropriate. Okay, now we're just going to cover some basic definitions. So hopefully by now you guys have an idea of what these definitions are anyways through the work that you guys do. I know that you work as MLTs, so we're just going to cover these pretty quickly. An acid is a substance that yields a hydrogen ion or hydronium ion when dissolved in water, whereas a base is a substance that can yield hydroxyl ions, denoted with OH negative. The dissociation constant or ionization constant, or K value, describes relative strengths of acids and bases. And the pKa is a formula that is calculated from that dissociation constant. So it is the negative log of the ionization constant and pH in which protonated and unprotonated forms are present in equal concentrations. And I've got a little note down here that strong acids equal pKa less than 3, and strong bases have a pKa of greater than 9. So you'll see that I have little notes in red here. These are just kind of little carrots of information that you might want to write down. They might be helpful in your understanding. And they also kind of just help you follow along with the lecture so that you're actively writing things down rather than just blindly listening to me if you're listening. A buffer is a combination of a weak acid or weak base and its salt, and this is a system that allows or actually resists changes in pH. And we're going to talk about buffers in a lot of depth today. And why is that? That's because what we're talking about is this homeostasis that our body carries out. And part of homeostasis is making sure that we have a good acid-base balance, and buffers are going to help us to achieve that. So, like I said before, acids are chemical substances that can donate protons, or you might also see protons notated as H plus ions in solution, and a strong acid is going to readily give up that proton. A base is what's going to accept the proton from that acid, and strong bases are going to readily accept that proton. So pH is essentially just a measure of those protons, or of those H plus ions. And as you guys already know, a basic pH is any pH that's greater than 7. Neutral pH comes out at exactly 7, and acidic is anything that goes below 7. And pH is a calculation that is the negative log of hydrogen ion activity. And so that's the equation that you see here, the negative log of the hydrogen ion activity. So because we're talking in logs, that means that a decrease of 1 pH unit. So if we go from 7 to 6, that actually represents a 10-fold increase in proton activity. And you can see that there is a table over here that kind of shows you what that really means, what the concentration of the hydrogen or hydroxide ions are, and how that relates back to our pH. So again, like I said, this is important because our body maintains homeostasis. It doesn't want our pH to change much. The body really can't tolerate the pH to really change a whole lot. And so because of that, there's got to be a mechanism in order to keep the pH in the same range, more or less. If the pH gets totally off whack, and by totally off whack, I mean just by maybe a couple tenths of a pH, this can really start to lead to alterations of cells. And in doing so, chemical reactions are not going to perform properly, and that's going to lead to alterations in consciousness. You could get neuromuscular irritability, tetany, and more serious implications including coma and death. So this is something that your body takes very seriously. The range that we're going to be dealing with for the pH, we're mostly going to be looking at the arterial pH. You will also, as a tech, be running venous blood gases, but arterial is kind of the basis that we use for learning, and then venous is off of that. So the arterial range is a pH of 7.35 to 7.45. So that's a very small window for us to get the pH right. And the way that we resist changes in pH within our system is through the buffering capacity of blood. So this is where buffers start to get really important. There are also other mechanisms in which we kind of keep all of this in balance, and we'll talk about those as well. They go hand in hand with the buffering capacity of blood, and they all work together. And that includes some respiratory mechanisms as well as renal regulatory mechanisms. Okay, so this just kind of goes over what acidosis is. Again, if something is acidic, that means that the pH is going to be lower. If something is basic or alkalotic, that means that the pH is going to be higher, and that's what we see here. Okay, so just to touch on some of the buffer systems, the most probably important buffer system that you're going to learn about today is the bicarbonate carbonic acid buffer system. And these are the chemical formulas for each of those, just so that you can start to become familiar with them. So bicarbonate is HCO3 negative, and carbonic acid is H2CO3. We also have what are called the hemoglobin and phosphate buffer systems. There are proteins that are able to act as buffers, the most well-known one being albumin. And then the kidneys also have a way of secreting acid in the form of ammonia in our urine, which is going to help to decrease the amount of protons or the acidity in the blood. Now, like I said just previously, we also have pulmonary and renal systems that work together. So pulmonary, meaning breathing in and out. As you guys know, when you breathe out, you are releasing CO2. And when you breathe in, you are taking in oxygen. So if you wanted to get rid of a lot of CO2 within your system, you would increase your respirations and really try to blow off this CO2. If you wanted to, let's say that you were more basic or alkalotic conditions, then in order to retain CO2 or make things more acidic, then your respirations would decrease. So you would take more slow, deep breaths. The renal system has ways of retaining HCO3, and that's going to increase our pH. Or, like I said before, excrete acids in the form of NH4 in order to decrease the pH. Okay, so again, we're just gonna be focusing on that first acid-base regulatory system that we talked about with bicarbonate and carbonic acid. So, like we said before, all buffers consist of a weak acid and its salt or conjugate base. So here I've just labeled this really important chemical equation. This is super, super important, and you should remember it. I've just kind of labeled who our weak acid is, and that would be the H2CO3, as well as our conjugate base, which would be the HCO3 negative. So just a regular old buffer system. And this buffer system is important for 3 reasons. So you can see that the equation has these like double arrows that are able to go both directions. And that means the body can kind of move this equation in either direction depending on what it needs. So one thing that H2CO3 as the weak acid can do is it can dissociate into carbon dioxide and H2O. And that allows us to breathe off some of that acid, right? So we are able to breathe off CO2 as well as the protons that come along with it as water. And that's just something that we do in our everyday life. We're breathing out CO2 and H2O. And again, you can change that CO2. You can change how much you are respiring. You can breathe faster, or you can take those low, deep breaths, depending on whether you want to retain CO2 or get rid of it. And again, we can alter the HCO3 conjugate base concentration with the kidneys. We can choose to retain that HCO3 if we have especially acidic conditions. Okay, so we had talked about earlier in the objectives the Henderson-Hasselbalch equation and how we can use that in order to assess a particular buffering system. So this is the Henderson-Hasselbalch equation. And basically what we can do is we can eventually just plug in our conjugate base and our weak acid into this formula, what the concentration of those are, and then we can assess our acidity or alkalinity using that equation. So a term that you might see here that may or may not be super familiar is the pKa. And I think there's actually a typo in you guys' notes, so you're going to want to put a little A there. The pKa is basically just a constant that relates back to acid dissociation. And the only thing I really want you to know specifically about the pKa is that the pKa of the bicarbonate buffering system is 6.1. So if we were to plug something into the Henderson-Hasselbalch equation using that really common buffer system that we're going to keep talking about, the bicarbonate carbonic acid buffering system, the pKa that you're going to plug in that case is 6.1. Okay. So these slides, you'll find that they kind of repeat themselves, but hopefully that shows you what is really important for you to know and understand in order to be successful with blood gases. So here we just break down that bicarbonate, or HCO3 negative, is the most important buffer system of plasma, this bicarbonate carbonic acid buffer system. And it is regulated by two separate systems in the body. So not just the respiratory system, but also the renal system. And this ratio here of the concentrations of HCO3 over H2CO3, so the weak acid and its conjugate base, we like to keep that ratio at 20:1. That's really important to note. 20:1 is where we like to keep that ratio, so more base than acid. Now, if you have even more base added to this particular ratio, let's say that you have an excess of HCO3 and you have less H2CO3, what's going to happen to the pH if you have more base? The pH is going to go up, right? And so thus, with the pH going up, we also see that the protons are going to decrease, right? And if we have decreases, this should say alkalosis. If you have decreases in alkalosis, that means that you have less base, right? So if you have less base, that means that you're going to be more acidic. And so your pH is going to be what drops, and your protons are going to increase, because the more acidic something is, the more protons. Okay, and so depending on what causes this increase or decrease, either the lungs or the kidneys will respond in order to restore that 20:1 ratio and to keep the pH within normal limits. And what are the normal limits? 7.35 to 7.45. And you can see that outlined up here too in this nice little diagram. Okay. So we've talked about the bicarbonate carbonic acid buffer system a little bit, and we will talk about it a little bit more, but now that you kind of know the basics and you understand that these can be adjusted in order to maintain that narrow pH window of 7.35 to 7.45, let's talk about a couple of other components that can act as buffers in our system. Hemoglobin is one of them. So hemoglobin is what is the major buffer of red blood cells. So it's going to actually be present in red blood cells, as you know hemoglobin is. And the component of hemoglobin that allows it to act as a buffer is what is called an imidazole group. And that is found in the histidine groups within hemoglobin. So the special thing about these imidazole groups is that they really like to bind protons, which is great because if you have a high amount of acidity, you need something to bind to those protons in order to keep them from disrupting the pH of our system. So if you have any changes that are going to result in a dissociation of hydrogen and you get free protons, we want the hemoglobin to bind those protons. Now, what else does hemoglobin bind? I'm sure you know this. Hemoglobin binds oxygen as well in the form of O2, right? That's its whole goal, really, is to transport oxygen throughout the system to tissues from breathing in the oxygen in our lungs, right? And then the heart is going to pick up that oxygen, distribute it through the blood, and then our tissues are going to benefit from that oxygen distribution. So that's something to kind of take note of here, of how hemoglobin's acting as a buffer. As the O2 binds to hemoglobin, now the protons are going to be released. If the hemoglobin lets go of that O2, now it's going to have room for the protons, and the protons are going to bind the hemoglobin. So that's just something to kind of take note of, is that hemoglobin is not acting as a proton carrier, or it's not holding on to protons as long as oxygen is present on those red blood cells. It's only after the O2 has been released that those protons are going to be picked up by the hemoglobin. Okay. We also have what's called the phosphate buffer system, and this is a very minor buffering system in the blood. You don't need to know much about this other than really the fact that we have protons that are secreted into the tubular lumen by the sodium-hydrogen exchange mechanism that we've previously learned about. And what happens is those protons react with the HPO4. And so as they're doing that, the HPO4 is going to accept those protons or those hydrogens. And when it does that, it's going to convert to H2PO4, which is the acid in this particular equation. So like we had talked about buffers before, we have our conjugate base. That's going to be our HPO4. And then we have our weak acid, and that would be the H2PO4 that's accepting or that's got the 2 protons. This is then excreted, but it doesn't significantly decrease the urinary pH. That's why we say it's just a minor buffering system of the blood. But it does get some of the acids excreted out of the body through the urine. So that's just one way that we can get rid of excess acid in the body, is to excrete it out with the urine. Like I said before, we also have some proteins that are used as buffers. We'll focus mostly on albumin, and you're going to see another common theme here. So albumin is a protein that can be found in the plasma, correct? And it has 16 imidazole groups from histidine. So it acts actually pretty similarly to hemoglobin in that its imidazole groups are attracted to those protons. So that's what they have in common, and they're going to bind or release protons very similar to the way that hemoglobin does, and that's going to help it to act as a buffer. Okay. So just really quickly, we're going to touch upon this just so that you kind of know why this is so important. And then we'll go back into the acid-base balance. So just really briefly, some of the acid-base disorders that we're going to come across are usually characterized or categorized into acidosis or alkalosis. So if you have acidemia, that means that you're carrying excess acid or proton concentrations in the blood. So your pH is going to be lower than the reference range, right? Because we have excess protons, that means it's an acidic condition, and according to the Henderson-Hasselbalch equation, that means that our pH is going to be lowered, and it's going to be lower than the reference range. You can also have, excuse me, alkalemia or excess base, in which case the pH is going to be greater than the reference range. So greater than that reference range of 7.35 to 7.45. You can have what is called non-respiratory acidosis, and this is where you have a decrease in bicarbonate, and that results in a decreased pH. So it doesn't necessarily mean that you are breathing or that you're not getting rid of enough CO2, but you actually just have a decrease in something basic, and that means that if you have less base, your pH is going to drop. You can have respiratory acidosis, which means that you've got a decrease in the ventilation through your alveoli, and we'll go through how the alveoli ventilate here in just a little bit. But that decrease in ventilation is going to cause you to not be able to blow off that CO2 as adequately, and we like to think of CO2 as an acid, so that's going to result in respiratory acidosis. Your pH is going to be lower than the reference range. Let's write that down. pH is going to be less than reference range. Just as we can have non-respiratory acidosis, we can also have non-respiratory alkalosis. This means that we have an increase for some reason of bicarbonate, which is going to cause an increase in pH as our bicarbonate is basic. And then we can have respiratory alkalosis as well, which means you have an increase in your alveolar ventilation. And this is going to cause you to get rid of too much CO2. So we want to get rid of some CO2, but if we get rid of too much CO2, then that's going to result in creating a basic condition. If we get rid of too much acid, that's going to raise our pH. So let's just write that down. Our pH is going to be greater than the reference range. And as we have briefly described before, the regulation of this acid-base balance can also be acted upon by the lungs and kidneys. So we have our buffering systems, and then we also have our lungs and kidneys. So if you haven't figured it out by now, ventilation, breathing in oxygen, breathing out CO2, is going to affect the pH of the blood, and that's because we think of CO2 as an acidic component. So CO2, or O2, as we breathe it in, it diffuses from the alveoli into the blood, and then the CO2 is going to diffuse into alveoli from the blood and be eliminated via ventilation. And we'll look at that more closely through a figure here in just a minute. The result of that is a minimal change in the proton concentration between venous and arterial circulations. So that's what's really important, right? Is we want to be able to exchange those protons from one carrier, if you will, to another without causing a huge change in those proton concentrations that our body is going to notice that's going to result in a pH change. The kidneys, on the other hand, are going to do this a little bit differently. The way that they're going to regulate the acid-base balance is by excreting acids or bases, and I should say, or retaining bases, such as HCO3. That's something that our kidneys are known to do as well. So the HCO3 can be reclaimed from the glomerular filtrate, and that's going to prevent excessive acid gain in the blood from loss of HCO3 in the urine. So if we're losing too much base or HCO3 out of our urine, that's going to cause an acidic condition within the blood. So what we can do is we can, well, I guess we don't tell them, but our kidneys automatically decide that it's a good idea to just start retaining some of that HCO3, retaining some base to resist acidic conditions. Okay, so I've said this before, but something that you should be kind of putting together by now is that you can think of CO2 as an acid, and you can associate it with respiratory imbalances because you're breathing out CO2. Similarly, you should be thinking of HCO3 or bicarbonate as a base, and this is something that you're going to want to associate with metabolic imbalances. Why? Because our kidneys have the ability to excrete or retain HCO3 in order to minimize major pH changes. So that one, anything related to the kidneys, we're going to refer to as a metabolic imbalance. Anything related to breathing in and out, naturally, is going to be a respiratory imbalance. Okay. So let's take a look at this pulmonary system regulation and how that kind of works. So again, here comes this really important equation, right? This is the bicarbonate carbonic acid buffer system that we just keep coming back to. So if you have in the middle, you have your weak acid, the carbonic acid, and then you've got your double-sided arrows, meaning that the equation can go back and forth depending on what the body deems necessary. Your weak acid can either dissociate into a proton and bicarbonate, or it can dissociate into CO2 and H2O, CO2 being an acid that we're going to breathe out. Okay. So if you have an acid increase, if the system starts to contain too many protons and our pH goes down, then what's going to happen is we're going to go here and our protons are going to combine with bicarbonate. Those are going to create this weak acid. That weak acid is then going to dissociate and become CO2 and H2O. And then we can release that CO2 through our respirations, and that should help us to get rid of some of the acid that has been increasing within the system. I hope that makes sense. If you have an alkali increase or your pH is going up, one way that the body can compensate for that is to retain your CO2. So CO2 is acidic, right? And things are getting too basic. So what can we do to alter that pH? We can start to retain some acid. So to do that, your body's going to know that it needs to respire less. So you're going to decrease your breathing activity. Okay. How does the renal system regulate? Again, we've briefly talked about this. The kidneys can choose to retain bases or they can choose to excrete more acid. So if they're going to retain the bicarbonate, if you happen to need more basic conditions, if they're going to retain bicarbonate, that's going to happen from the proximal tubule of the kidneys. If you need to excrete more acids, that's going to happen via the distal tubule. And so that can happen in a couple of different ways. We can secrete protons specifically by using the bicarbonate carbonic acid buffer that we've been talking about so much. We can combine protons, and we're going to look at this actually on the next slide, but it never hurts to go over twice. You can combine protons with NH3 to make NH4, which is secreted or excreted out of the urine. And we can have an H2PO4 phosphate buffer as well. So that was what we talked about previously, where we had HPO4 and we were having it pick up a proton to make H2PO4, which is then excreted out of the urine. So both of these can be excreted. Okay. So, like we'd previously discussed just now, you can use ammonia as a way to release acid out of the body by excreting it from the urine. Okay, so now we're going to talk about the distribution of gases in the body. So we're kind of moving now from exactly how this acid-base homeostasis works into how do we really distribute oxygen and CO2 throughout the body, and how do they kind of keep each other in check. So we're going to look at the transport of oxygen, we're going to look at the transport of carbon dioxide in the blood, and then we're going to look at the hemoglobin oxygen dissociation curve. So to do this, to understand this, the first thing you need to understand is about partial pressure. So partial pressure, anytime that you see like a PO2 or a PCO2, all that's telling you is it's the partial pressure. And this just comes into play when you're dealing with gases. So when more than one gas is present in a container or within the body, which of course is always going to happen. You're never going to have 100% of one gas and 0% of the other. The pressure exerted by one gas is called the partial pressure. So this just kind of shows us the composition and partial pressures of gases in a dry air atmosphere. So these are just any number of gases. Okay, so now that we kind of know what the partial pressure concept is, and we can understand that partial pressure is really just a way of expressing how much of a particular gas is, how much pressure a particular gas is exerting in a container or a body, we can start to apply that to how we transport CO2 and oxygen throughout the body, and how that transport really depends on partial pressure. A lot of this is going to rely on the fact that gases travel from high concentrations to low concentrations. Pretty basic concept there, and you'll see how that applies here. So I left a little box here saying that we were going to draw something out in the video lecture, and as promised, we are going to draw something out, but after thinking about it, probably is not going to fit into this little box. If you can fit it into this little box, that's awesome. I will not be able to. So I'm just going to add a page to the notes, and I'm going to draw it out on a full page or maybe even a couple of pages. And I would really highly suggest that you draw this along with me or follow along with me, but probably draw it along with me, because this is really going to help you to hammer in these concepts. It really helped me to understand them better, to be able to use them in the lab. So I would really recommend that you draw this with me. So if you don't have a blank piece of paper, you're going to want to get one now. And you can pause the video and grab a blank piece of paper or a couple pieces of paper, some colored pencils if you have it, and let's get to drawing this thing out. Okay, so we're going to start with a blank slate here, and we're just going to write the definition of partial pressure up at the top. So partial pressure, as discussed previously, it's very similar to the concentration of a gas, the amount of pressure that a gas is putting on a system. The higher the partial pressure, the larger amount of gas present. So it's proportional to the concentration. Okay, so if we were to draw a guy, and forgive my cartoon drawing skills, if we were to draw a very serious looking guy, I don't know, with apparently an egg-shaped head. Oh, that wasn't so bad. If we were to draw a guy and we were to draw his respiratory airway down into his lungs, and his diaphragm. Okay, now we can start to talk about how air moves around. So let's talk about what is present in our atmosphere, what we're breathing in terms of the partial pressure of oxygen and CO2. So in the atmosphere alone, our PO2 is going to be around 160 millimeters of mercury, or mmHg. And this is the only time I'm going to write millimeters of mercury, just for the concept of saving space. But rest assured, any of the partial pressures that we're going to show on this diagram are going to be denoted in millimeters of Hg, or millimeters of mercury. So the partial pressure of O2 in the atmosphere around 160 millimeters of mercury, whereas CO2 is actually only around about 0.3. So that's what we're breathing in. And as you know, we're going to eventually transport the oxygen to our tissues to be used. Okay, I'm going to use some colors here to hopefully try to make this pretty clear. So as you can see, the PO2 is a lot higher in the atmosphere than the PCO2. Okay, so we're going to breathe that in. And it's going to go down into the lungs. Now, in turn, what are we breathing out? We're breathing out CO2, right? Mostly. We're also breathing out some oxygen. But CO2 is what we think of when we think of breathing out. So the partial pressure of what we are breathing out, we're actually going to have a PO2 of around 120. This is surprisingly high, right? And then we're going to have a partial pressure of CO2 of about 27. This is what we breathe out. So just kind of a quiz question, I guess, or a fun trivia question. If we're breathing out that much CO2, what's happening to it in the atmosphere? Because it's going from 27 to 0.3. What do you think is happening? I'm sure you've guessed it. The plants, right? The plants in our world are taking that CO2 and using it for their cellular respiration and in turn giving us oxygen. So they're taking the CO2, they're doing some stuff with it, which we're not going to go into today. And then from that CO2, they're making oxygen for us to breathe in. Okay, so let's keep drawing. So now that oxygen and CO2 that we are breathing in is going to go down into the lungs, right? And it's going to go into the alveoli. So I'm just going to draw kind of what a zoomed in picture of the alveoli would look like. And it's probably going to be pretty bad. Okay, so we've got some alveoli there. Each of these little individual circles being the alveoli. And then we've got the pulmonary artery, which is going to take deoxygenated blood and it's going to deliver it to the alveoli. So this is our pulmonary artery, and it's going to deliver deoxygenated blood to alveoli. Now when